Flowers are always been used as a symbol for conveying human feelings, isn't it? Do you think they are meant for it? No, not at all. They are the reproductive part of the plant. So, in this chapter, that is sexual reproduction in flowering plant, we will understand how flower transform into a fruit. So, welcome to the channel that is Biopulse, and this is Chandrika. Today we are going to start a new chapter, that is sexual reproduction in flowering plants, and this chapter having a very high mark weightage both in your theory session also and for your NEET session also. So let's get started. What are the topics we are going to cover for today? So we are going to discuss about structure of flower, in that female and then male reproductive part, especially stamen and then pistil, okay, and pre-fertilization event, post-fertilization event, after the pre-fertilization event, pollination and their types and agent for the pollination, adaptations in the flowers, how actually the fertilization occurs. What is fertilization means? The combination or else the fusion of the male and then female gametes. So in the angiosperms, what happens? Triple fusion will be there. Those all things we are going to discuss in upcoming sessions. After that, post fertilization events are there, and then special topics like apomixis, polyembryon and then parthenocarpy. So these are the main parts which we are going to cover in this topic. So we will start with the introduction part. All the flowering plants show sexual reproduction. That's what, that is angiosperms we used to call, and flowers are the morphological and embryological marvels and sites of what? Sexual reproduction. Whatever the flower is there, no, that flower we consider it as a morphological and embryological marvels, and it is a main site for what? Sexual reproduction, for continuation of the life. So due to several hormonal and structural changes, what happens? It results in the differentiation and development of the floral primordia. What is floral primordia means? See, whenever we talk about the stem, there will be a stem, apical meristem. Here there will be a small apical meristem area. Here what happens? In this area, due to the several hormonal changes, that there will be a differentiation and they will form what? Floral primordias. These are the floral primordias, okay, and inflorescence bear floral buds and then flowers. This is the inflorescence which I am showing here. See, what is the purpose of plant producing inflorescence? From the far away, insects or else pollinators, they can't see the flowers. If the flower is unique, only one flower is there in a plant means, that's what happens. There will be a bunch of flowers in the form of what? Inflorescence, so that they can attract the pollinators very easily. So that is the purpose actually the plant produce the inflorescence. So here in these all inflorescence, these small tiny they are the flowers actually. This is the inflorescence of onion, that is Allium cepa. So here this is the bud, and the bud is after the differentiation, it is going to form in the form of bud. Then next bud is going to bloom in the form of what? Flowers. So this is the actual stage. So flowers arise from stem meristematic tissue, stem apical meristematic tissue. Is that clear? That's good. Next, this is the structure of flower, a typical structure of the flower. When we talk about the parts of the flower, flower is having mainly two parts, that is vegetative part and then reproductive part. Again, vegetative part is divided into two types and reproductive part is divided into two types. Vegetative part will helpful for the protection, attraction, those all things, and then reproductive part are meant for the reproduction, that is sexual reproduction. So here in the vegetative part, First one is calyx, and unit of calyx is called as sepals. Second one is corolla, and unit of corolla is called as what? Petals. What is unit? This only one thing if I ask, this is called petal. Whole overall whorl if I ask, it is called as what? Corolla. Only one thing if I am asking, it is called as sepala. Whole whorl is called what? Calyx. Got it? See, this is calyx, this is corolla, this is androecium, and this one is gynoecium. So here male reproductive part is said to be a androecium, and its units are said to be stamens. These all are stamens together called as what? Androecium. And the female reproductive part is said to be a gynoecium, and its unit is said to be pistil, or else you can also call it as a carpel. So here is the structure. This is the sepal, and this is the receptacle we used to call, and stalk of the flower is called as what? Pedicel. Stalk of the flower is said to be pedicel. First outermost layer is calyx, next comes corolla, next comes androecium, and then the innermost will be of what? Gynoecium. So here when we talk about in detail, one by one we are going to talk about in detail of all the whorls. So here first we will discuss about the vegetative parts, and they are also called as accessory whorls, whereas reproductive parts are said to be necessary whorls and vegetative parts are said to be accessory whorls. Why they are called as accessory? Because for example, if I say in our life, laptop, mobile, these all things are said to be as accessory tools because they are needed, but they are not compulsory to live, isn't it? But air, food, water, these are necessary things because we depend on them to be alive, isn't it? Same like that for the flower or else for the plant, the vegetative parts are said to be accessory whorls because they are necessary, but they are not directly involving in the reproduction. So how many accessory parts are there? Two, sepals and then petals. Here sepals are the whorl units of calyx and petals are the units of corolla. Sepals usually they will be green. This is the sepal, okay, and it is leafy like structure that protect the bud before it opens. It protects the bud. See here, this is the bud. It is protecting the bud before it is opening. So sepal is meant for the protection. Next, when we talk about the petals, they are very bright in colors. Whatever the flower we are going to be attracted nowadays, no, they are due to the presence of the petals. Green, white, yeah, of course many of the flowers are green in color also. Pink, red, white, etc, etc. Many of the colors are there that we are going to attracted by, isn't it? Those all attraction is due to the presence of the petals, and they are often scented to attract pollinators like bees and then butterflies. Okay, now why they are colorful? They are colorful because they want to attract the pollinator for the pollination. So this one is the petal and this one green color is what? Sepal. Next, reproductive part. So this is called as what? Essential whorl. Why? Because it is must be there for the reproduction. They must be there in the flower for the reproduction purpose. That's what they are said to be essential or else necessary whorls. So what are the essential or else reproductive parts? Male and then female organs that enables the fertilization. So here male organ is said to be androecium and its unit is said to be stamen. Okay, part of androecium. Female is said to be a gynoecium and its unit is called as what? Pistil or else carpel. Here this is the male reproductive part and this is the female reproductive part. Let me zoom. See, this is the female reproductive part, whereas this is the male reproductive part. Male reproductive part, again it has two main parts. This is the anther and this one is the filament. I will explain in detail. And this one, the female reproductive part, has three parts: stigma, style and ovary. This is the stigma, landing platform, and then there will be a hollow portion which is said to be a style, and there will be a bulged portion which is having the ovules. That portion is said to be ovary. So male and then female parts, reproductive part, are androecium is the male, gynoecium is the female. Androecium has two parts, anther and then filament, whereas gynoecium has three parts: stigma, style and ovary. Is that clear? That's good. Next, so in the male reproductive part, it is having anther and then filament. Two main parts are there. Anther, which is main part, which is produce pollen grains. Pollen grains are the male gametophytes which carry the gametes. Okay, so pollen grains are the male gametophyte. Filament, it is a slender stalk that holds the anther. See, this is the stalk which holds what? Anther. This is called what? Filament. And when we talk about the female, it has three parts: stigma, style and then ovary. Stigma, which is this portion which is very sticky surface that captures what? Pollen grains. And whereas this style is tube like, connecting stigma to the ovary. This will be tube like which connects the stigma to the ovary. So whenever the pollen grains, which here these are the pollen grains, whenever these pollen grains are going to attach here, they are going to grow a tube like, that is called as pollen tube, and which carry the pollen male gametes, which will be up to the ovules. So stigma is the landing platform, whereas tube, which is going to connect the stigma and then ovary for the growth of the pollen tube, and whereas ovary, which contains what? Ovules. Okay, after fertilization, this ovary itself becomes fruit, and ovules, whatever the inside small tiny portions are there, no, these are the ovules. These ovules become seeds after fertilization. This is actually the female gametophyte which is present. Ovules have female gametophyte which is said to be a embryo sac. Later on you will come to know about that. Next, let us talk about in detail what actually the androecium is. Androecium is the male reproductive part, isn't it? Androecium consists of whorls of stamens, whorls in the sense whole thing, stamens representing the male reproductive organ. This is only one stamen I am showing. Many stamens will form a whorl. Whole thing is said to be androecium, isn't it? When we talk about only one stamen, it has anther and then the long and then slender filament. Okay, so let us talk about filament first. A long and slender stalk called what? Filament. This is the filament. The proximal end of the filament is attached to the thalamus or petal of the flower. This proximal end, it might be attached to the thalamus or else petal of the flower. Okay, if I show you here, see, if it is attaching to the thalamus or else it may be attached to what? Petals. Okay, again there are many types, we will discuss about that also. The proximal end of the filament may attach to the thalamus or else they may attach to the petal of the flower. When we talk about the anther, the terminal, generally this one is the terminal portion. Okay, this one is proximal end. The terminal generally bilobed structure. Bilobed. Why it is called bilobed? Because it is having two lobes. This one is one and then this one is two. Each lobe has two thecas. Means if I am talking only one, it is having two thecas. This one has the two thecas, dithecous condition. And there will be a longitudinal groove runs lengthwise separated the theca. There will be a longitudinal groove separating the theca. There will be a groove. This is one theca and then this one is one more theca. Dithecous. Okay. This is one lobe and this one is one lobe. Let me draw here. This one is one lobe and there will be another lobe. In each lobe, that's what it is called as what? Bilobed. One and then two lobes. These are the lobes. And in each lobe there will be a theca. One theca, two theca. One theca, two theca. That's what it is called what? Two theca. That is dithecous. How many thecas totally? 4. But in each lobe it is having two thecas. That's what it is called, dithecous. Okay. This is the filament. This is the anther. Let me zoom. Give me a second. See. This is the connective which connects the two lobes. This is one lobe and then this one is one more lobe. This is one sac. Okay. That is said to be a pollen sac. Inside that what happens? There will be a small tiny pollen grains we used to call. See here. 1, 2, 3, 4. I can count them easily, isn't it? So these are the male reproductive part, pollen grains. Okay, this is what actually called as microphyta. These are the microsporangia. So how many lobes are there? 2 lobes. And in each lobe how many thecas? 2 thecas. This is one theca, this is one more theca. And inside the theca what they are having? They are having the pollen grains. Next comes, when we take the section, when we cut the anther in a transfer section, we will see the structure like this. So this is the anther. When we cut the anther, that will be showing you like this. It will be having 2 lobes. This is the one lobe and this one is the one more lobe. And each lobe is having 2 thecas. Okay na? And inside that theca, there will be a pollen grains. And this portion is called as what? Pollen sac, or else it is also called as microsporangia. So anther is a 4-sided tetragonal structure. 4-sided. 1, 2, 3 and then 4. 4-sided tetragonal. It is tetragonal bilobed dithecous. Okay. Consists of 4 microsporangia located at the corner. This one is one microsporangia. This one is second. 1, 2, 3 and then 4. It is having 4 microsporangias located at the corners. 2 in each lobe. So if I slit like this, this is one lobe and this one is second lobe, isn't it? So one lobe is having 2 thecas, one more lobe is also having 2 thecas. That's what it is said to be what? Dithecous and bilobed. The microsporangia develop further and become pollen sacs. When we talk about initial, that is when the anther is young, it is said to be a microsporangia. After the development only, they are going to call it as what? Pollen sac, which is bearing pollen grains. They extend longitudinally all through the length of an anther and are packed with the pollen grains. They are what? They are packed with the pollen grains. This is what the TS of anther. It may ask for 3 marks for your theory examination, and from the diagram session also for the NEET, many of the times they are asked the part labelings. Okay, I will explain you later in the next slides. See, just you must know in this slide, This is the lobe, one lobe, and this one is the one lobe, and two lobes are connected with a portion called as what? Connective. This is said to be a connective, which is having the vascular bundle. Once they ask this question also, connective in one connective it is called, and which is having the vascular bundles in the sense xylem and then phloem. Who is responsible for the food and then water supply for the developing anther means connective, because it is having the vascular bundle. Next, structure of microsporangium, only one. See here, it is going to be very neat. This is one lobe and then this one is one lobe. Each lobe is having two microsporangia, and this microsporangia again we are going to zooming out. Only this portion is zooming. Okay. A typical microsporangium appears near circular in outline. See, this is the near circular one. One sporangium, two sporangium, third sporangium, and then fourth sporangium. In this, only half portion is zoomed out here. Okay, and this is the connective. It is generally surrounded by four wall layers. Usually what happens, it is surrounded by microsporangium. When we talk, it is having four wall layers. Please keep in mind as in this way only. Okay, this is from outside to inside, and from inside to outside, like this. Okay, outermost will be epidermis, next will be endothecium, next middle layer and tapetum. This is the epidermis. Let me zoom. See here, this is epidermis, outermost single layer. Then next comes endothecium. Then next there will be a 2 to 3 layer of middle layers, and at last there will be a tapetum. This is the tapetum, which is having the multinucleated condition. This is the portion, tapetum portion, which you may expect most of the times many questions will arise from this. So outermost will be epidermis, next comes endothecium, next middle layer, then next there will be a tapetum. Epidermis, endothecium, middle layer, tapetum. After the tapetum, there will be a sporogenous tissue. Before forming the pollen grain, sporogenous tissue. That sporogenous tissue will differentiate in the form of microspore mother cells, which will give you microspores. Okay. The outer three wall layers perform the function of protection. When we talk about these three, which one? Epidermis, endothecium, and then middle layer, they are meant for what? Protection. They are meant for protection. When we talk about the tapetum, this is for the nourishment. Okay, so the innermost wall layer is tapetum, and it nourishes the developing what? Pollen grains. Whenever here the pollen grains are going to form, no, that innermost wall layer, that is tapetum, is going to nourish them by providing the food and then other supplements. So cells of the tapetum possess dense cytoplasm. Dense cytoplasm means cytoplasm is very active, and it is usually having more nucleus, not one, more than one nucleus. Usually each cell will be having only one nucleus, no, but here in the tapetum you can see multinucleated condition. That question already asked many of the times. So among them, which is having multinucleated condition, that is tapetum. Among these layers, which provide protection? Epidermis, endothecium, and middle layer. Among these layers, which is going to nourishes the developing pollen grain? Tapetum. Yes, I hope this is clear to everybody. Yes. Next, see here, clear cut division of the role is there. Epidermis is for the protection, protecting the outer layer. Next, endothecium, it helps in anther opening. Anther opening means whenever the anther is going to be mature after the formation of the pollen grains, that must be open, isn't it? Because that formed pollen grains must be exposed to the air or else must be exposed outside. So endothecium will helpful for the opening of the anther. Next, middle layer, it will be temporarily support and for the lateral degenerate. Later what happened, it is going to be degenerated. And tapetum, you also know, it nourishes what? Developing pollen. When we talk about the pollen sac, it is the site of what? Pollen grain formation. Here is the very clear cut division. See, this is blue color one is epidermis, outermost one. Next comes the endothecium, which is second layer, and then third one is middle layer, which is multi layer. See, two layers are there, 2 to 3 layers of middle layer there will be. And then again the single one, which is very important, which is called as what? Tapetum, which provides the nourish to the developing pollen grain. Here are the spore cells, that is microspore mother cell. In young anther, each microsporangium has sporogenous tissue at the center. This is the sporogenous tissue. Again there will be huge number of the cells which are said to be microspore mother cell. It consists of compactly arranged sporogenous cells. These are the sporogenous cells. Okay. Next, how actually sporogenesis is going to be happen? If I take only one, okay, everything will be same for other cells also. If I take only one cell, that cell is called as what? Microspore mother cell, that is MMC. Okay, when we talking about what happens next to this, the process is said to be microsporogenesis. Microspore is called as pollen grain and genesis is meant for the synthesis. So synthesis of the pollen grain is said to be what? Microspore mother cell. In the anther, this is the anther and this is the filament. In the anther, when we cut, it looks like this. This is having two lobes. This is one lobe. This one is second lobe, isn't it? Each lobe is having two thecas. This is one and then this is two. This is pollen sac, this is pollen. Inside that there will be a tiny cells which are said to be what? Sporogenous tissue or else MMC. After that what happens? They are going to mature to form a MMC. As the anther develops, the cells of sporogenous tissue undergo meiotic division to form a microspore tetrad. Okay. The microspores as they are formed are arranged in a clusters of 4 cells. The process of formation of microspores from a pollen mother cells, that is PMC or else MMC also you can say, through meiosis is called microsporogenesis. What it is? The process of formation of microspores from a pollen mother cell, that means from a pollen mother cell, formation of microspores is said to be what? Microsporogenesis. Through which? Through meiosis. Keep in mind, it takes place by the meiosis. Okay. As the anther matures and dehydrate, the microspore dissociate from each other and develop into pollen grain. What it means? See, this is the 1 PMC. Let me, okay, this is having the ploidy 2N. Okay, here first meiotic division will lead dyad condition. After the second meiotic division, you will be having tetrad condition. 4 cells will be there and they are going to call it as a tetrad condition, which will be having the reduced number n, because they are going to form a male gametophyte. in later on. After maturation, what happens? They are going to give you pollen grain. There is a lot of difference between the microspore tetrad and then pollen grain, because microspore tetrad is a prematured stage of the pollen grain, whereas pollen grain or else microspore we used to call it is a matured stage. How it is going to be matured? Once they are formed the tetrad, whatever the outer tapetum layer is there, no, this layer, it is going to produce a sporopollen on those all layers, those all cells, to form a pollen grain to be a stiff manner. That is maturation stage we used to call. After the anther maturation, what happens? Develops into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. Dehiscence means what? When this anther is going to be bursted. Here, from this portion, what happens? Anther is going to be opened up. After that, what happens? All the pollen grains which are formed here, they are going to come out. Now let us talk about each pollen grain. So from 1 mmc or else 1 pmc, how many pollen grains you will get? From 1 mmc we will get 4 pollen grains, isn't it? Yes, keep in mind this one. MMC is having 2n ploidy, and whereas pollen grain is having n ploidy. N. Next, let's talk about what actually the pollen grain is. Pollen grain represents the male gametophyte. Male gametophyte. Why it is called as male gametophyte? Because it carries male gametes, generally which is of spherical in structure, measures about 25 to 50 mm in diameter. When we talk about this diameter, it will be about 25 to 50 mm. This is already asked. What is the size of the pollen grain? 25 to 50 mm in diameter. This is the diameter, and it has two layered wall. And even it has two cells also. The outer one is called exine and then inner one is called intine. And how many cells it is having? Two cells, tube cell and then generative cell. Okay na? Wholly this overall thing is called as pollen grain, which is having two layers, wall layers. Outermost is called as exine and then innermost is called as intine. How you will remember this? Exine, exit, okay, outer one. Intine, inner one, okay, like that you can remind them. So these are the many of the types of the different archaeological structure of the pollen grains. This will be due to the presence of a material which is very special to the pollen grains, that is called as sporopollenin. See, this pollen grain is different, this one is different, this one, all they are different. They look spiny, they look hold, they may be smooth somewhat, like this. These all are different kind of the species is pollen grains. For each species, the pollen grain exines sporopollenin structure will be different. We'll learn about that. When we talk about the exine, this is very important. This is overall whole thing is called as pollen grain. The outermost layer, exine, we are going to discuss now. This is a hard outer layer made up of what? Sporopollenin, which is very important. The most resistant organic material till today. No enzyme, no temperature, no acid, no alkali can destroy this sporopollenin. Do you know that? Yeah, that's what it is said to be. What? The most resistant organic material. Which one? The sporopollen. When we talk about the exine is made up of what? Sporopollenin, which is, it can withstand high temperature, strong acid and alkali. No enzyme can degrade what? Sporopollenin. which is present in exine. Pollen grain's exine has prominent apertures. Apertures means this one. This is the aperture. Called what? Germ pores, where sporopollenin is absent. This is the only area where this sporopollenin absent. Now tell me in comment box, which is the area where this sporopollenin is absent. I will wait for your answer. Yes, the area where the sporopollenin is absent, that is the germ pore. Now tell me, in this pollen grain, how many germ pores are there? Please comment your answer in comment box. Now, next, the pollen grains are well preserved as fossils because of the presence of the sporopollenin. Due to the presence of the sporopollenin, we can preserve the pollen grain for the many of the years in a pollen bank. Next, let's talk about the inner one, that is the intine portion. Intine is the inner wall of the pollen grain and is called what? Intine. And it is very thin, consists of layer made up of cellulose and then pectin. This is also important. Pectin and cellulose. Outer one, exine, is made up of sporopollenin. Intine is made up of cellulose and pectin. Please keep in mind. As I said, pollen grain has two layers, that is exine, which is made up of sporopollenin. This sporopollenin, which is withstand for high temperature, strong acid, alkaline, and no enzyme can degrade this sporopollenin. Because of that, what happens? We can store the pollen grains in a pollen bank, and where the sporopollenin is absent, that area is said to be what? Germ pores. And when we talk about the intine, it is the innermost layer and made up of cellulose and then pectin. Let's talk about the cells of the pollen grain. See, in the mature pollen grains, it contains two cells. As I said, one is vegetative cell and one more is generative cell. See this one, the large one is said to be vegetative one, and then small cell is there, no? This one, yes, this one is said to be generative cell. Vegetative cell is bigger, has abundant food reserve and large irregular shaped nucleus. This is already asked. See, if you observe this nucleus, let me zoom. See, if you observe this nucleus, this is the tubes, this is, yeah, of course you can call it as vegetative cell and then tube cell also. This is the tube cell, which is having the tube nucleus, which is not regular in shape, which is irregular in shape. Shape is not regular. When we talk about the generative cell, it is smaller, which is embedded in the tube cell, and it is having the prominent nucleus, isn't it? The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and then nucleus. This is the spindle shape. Generative cell's nucleus is having spindle shaped cytoplasm and a nucleus. Generative cell is spindle shaped, whereas tube cell is, or else vegetative cell is bigger one, and it has a abundant nucleus and then irregular shape of the nucleus. This is irregular. In over 60% of angiosperms, pollen grain are shed. Shed in the sense they come out of the anther at a two-celled stage. In 60% of the angiosperm. Remaining 40% species, the generative cell divides mitotically to give rise to two male gametes before pollen grain are shed. That's what it is called as three-celled stage. See, don't get confused here. If I ask you, this is the pollen grain, which is having the one irregular nucleus and one more is like this. This is two-celled stage, which you can see almost in 60% of the angiosperms. if I'm asking this generative cell and this is the vegetative cell, in some 40% of the angiosperm, what happens inside the nucleus only, inside this cell only, this one generative cell, again it is going to be divided to form two tiny cells which are said to be male gametes, and this one will be the larger and it's a nucleus, irregular nucleus. Let me rub. I will draw once again. See this one generative. In this generative, which division will occur? Mitosis. Mitosis in this, already this is n, okay, n in number, this is also n in number. N number of generative cell will undergo mitosis, will give you, this is pollen grain, isn't it? Inside that there will be a two generative cells, or else we used to call them as a male gametes, and there will be a one vegetative cell. So tell me, this is one and then this is two cells. Here this is one cell, the bigger one, inside that again two cells are there, so totally three. This kind of pollen grains are going to be shed in almost 60% of the angiosperms, whereas this kind of cells is going to be shed in remaining, that is almost 40% of the angiosperm species. Okay, that means they may be shed in two celled stage, they may be shed in three celled stage. If two celled stage, it will be having two cells with, that is vegetative and then generative. If I am talking about three cell, it will be having one vegetative cell and two generative cells. Pollen grains have to land on a stigma before they lose viability. Viability means capacity to form a pollen tube like that. Okay, in some cereals, like in cereals such as wheat and pollen grain lose viability within 30 minutes. What is the viability of the wheat pollen grain? That is 30 minutes of the release. Imagine once the wheat is released its pollen grain out, it must go and reach to the stigma within 30 minutes. If it can't, then it lose its capacity to grow. That's what the viability is, 30 minutes. In some members like Rosaceae, Leguminosae and Solanaceae, they maintain viability for several months. So viability may be within a seconds also, within a minutes also, within a months also. So here I am showing this is the wheat anthers. Next, economic importance of pollen grain. What is the economic importance? It has very much economic importance, and many of the questions will ask from this also. Pollen grains are very rich in nutrients. They are very rich in nutrients because it intine is made up of cellulose and then pectin, isn't it? Yeah. Pollen tablets are used as a food supplements. Pollen tablets and syrups increases the performance of the athletes and race horses. As you know, in the market there are many availability of the pollen tablets. What is pollen tablets actually? They are going to remove the exine and then they are taking out the intine, and they make syrups and then tablets of that content, and they are going to use to increase the performances of the athletes and then race horses, to increase their energy. And it is possible to store pollen grains of a large number of the species for years in liquid nitrogen. Keep in mind, liquid nitrogen at minus 196 degrees Celsius, not this 196 it is 196 degrees Celsius. Okay, so here when we talk about this importance of the pollen grain, that there is lots of economic important. So the pollen grains where we used to store, that is said to be pollen bank. Okay, and we used to store the pollen grains in a liquid nitrogen which is of minus 196 degree Celsius. This is not 190, that is 196 degree Celsius, minus, keep in mind, okay, minus 196 degrees Celsius, and that area, that place is called as what? Pollen banks. Next, pollen grains of some plants, that is like parthenium and carrot grass, are allergic for some peoples. It leads to chronic respiratory disorders. Parthenium and carrot grass will lead to asthma and then bronchitis in some of the peoples because they are having the allergics. So here are the some pictures I have given for you. This is the pollen, bee pollens, okay, and can be made into the different kinds. This is the pollen, bee pollen tablet, bee pollen powder, and this is also a tablet, and this is the syrup, I can say. So this is the tablet. So for today we will end up here. For the next class we will continue with the lecture number 2. So this is the lecture number 1. We will continue with the lecture number 2 for next class. Thank you. Bye bye. Please, if you like my videos and if you understand whatever I am going to teach you, like my channel, subscribe and share the videos. Thank you everyone.